have helped you in lab had you had class on Monday. But you didn't, so you have to learn it now. Okay, so membrane, plasma membrane. You look at this and what do you see? Light, dark, light, dark, light, dark. And what you're actually looking at are two cells that are attached together with space in between the cells. Looks pretty dark there. That's telling you that the cells are attached together by proteins, which are picking up the stain, and that's why there's the dark structures there. Then you run into two dark lines with a very light line in between. And same thing over on this cell side. Dark, light, dark. That's actually two structures that are associated together to make something that looks like 3 layers. Actually, only 2. So what's labeled as dark is the phosphate head of your phospholipid molecule. It's going to pick up more of the stain. So you're going to have a charge up here, fatty acids there that repel the stain, another phosphate down here that will pick up stain, and fatty acids that will extend into the center. These will appear light, these will appear dark. So actually 2 layers that make something that looks like 3 layers. So phosphate heads with the charge up here gonna associate with the hydrogen of the water. And this is going to associate with the hydrogen of the water. This is going to be on the intracellular side because I decided that's going to be ICF. And this is going to be on the ECF, extracellular side. So you got water inside your cells, you got water outside your cells. And we're going to learn about that plasma membrane and what that plasma membrane is doing. It's forming a partition. A partition is a barrier. This is a selectively permeable barrier. It lets some things across and it excludes other things. We want to keep the inside of our cell. We want to keep things inside the cell. So part of what we're going to do is form across this plasma membrane a partition for our electrolytes. We're going to have potassium in high concentration. So you're lazy, you don't want to write high concentration. Make a big K plus. High concentration inside, small K plus means low concentration outside. You're also partitioning sodium. Sodium's going to be in high concentration outside, and it's going to be in low concentration inside. So we started partitioning the ions. Other things are also going to be partitioned. You want to keep your nucleus inside your cell. You want to keep your other organelles inside your cell. You don't want to be chasing them down to bring them back to do the work for you. You want to partition them and keep them inside the cell. And you want to keep certain things out of the cell. So that's what your plasma membrane is there to do, is keep this partition going. On your printout from La Lima, you have the picture of the phospholipid. I don't know where it went, but it disappeared. Maybe I accidentally deleted it when I was condensing things for presentation. But anyway, I drew it for you on the chalkboard, on the whiteboard. The head will be negatively charged. That means that side of the molecule is polar. It associates with water. Another term for associating with water is it's going to be philic. It means it likes. It's hydrophilic because it likes water. This doesn't like water. That's why all the fatty acid tails go to the center of the bilayer. These are hydrophobic. So these don't like water. You can hydrophobic and hydrophilic. You also can describe these components of the phospholipids in an alternative fashion, but it means the same thing. This is hydrophilic, but it's lipophobic. This is hydrophobic, it is lipophilic. Having one molecule that has two types of interactions, this is called amphi. Amphi means both characteristics of liking and not liking. What you put here, it could be, you could put amphiphilic, means it has a characteristic that this molecule likes water and it likes lipid in each part of it. So this could be philic. You could say it's amphiphobic because this doesn't like lipid and this doesn't like water. So it has characteristics of both. And then just to make it even worse, sometimes they call it amphipathic. And that means, again, that you have characteristics of both liking and not liking. So the advantage of these phospholipids and using phospholipids is you can make the whole plasma membrane, 95% of the plasma membrane is out of these phospholipids. And then you can insert other things into these phospholipids. The phospholipids the fatty acid tails may pack very closely together, but sometimes there's a double bond here that's going to kink one of those tails and push another phospholipid a little further away. So that gives you a fluidity to the membrane so things can move in the membrane. And you need to be able to move things in that membrane. Also, your body will insert cholesterol into the fatty acid tails. And the cholesterol molecule will push some of those fatty acid tails apart. Again, to create the fluidity for the movement of things in the plasma membrane. So, can skip this slide, I think. Well, here the kinks in the tails increase fluidity, and then on the, not that slide, here you can see the cholesterol in the membrane. They're also pushing the fatty acid tails apart, so that's increasing fluidity as well. So you need to be able to move things through that plasma membrane. Here we've got not a true cell, but if we take plasma membrane and throw it into water, it's going to form these little balls. Some of the water is going to be trapped inside, and then the rest of the water is going to be on the outside. And so we've now modeled a cell for us that we can play around with. The Phosphates will always face the water, and the lipid will always be sequestered inside, away from the water. You can skip that. Okay, so we're partitioning, and we've got now an intracellular fluid and an extracellular fluid with a hydrophobic layer, or we got lipophilic outside and hydrophilic outside, hydrophilic inside with lipophobic in between. The fact that this layer here is lipophobic does not mean that water can't get through it. Water does actually diffuse through the bilayer, doesn't diffuse that quickly, but the hydrogen on the water is going to be attracted to the negative charge on the phospholipid. And when the molecule gets close enough to the phosphate, it's going to be repelled by the negative charge on the oxygen, and it tumbles into the plasma, into the hydrophobic part of the membrane. And water wants to get out of that. It doesn't like to associate with the nonpolar molecules. It could tumble back out, but it's already tumbling in, so its momentum is to go in. So as it's tumbling in, it gets attracted by the hydrogens to this phosphate and keeps tumbling inwards, and then it's repelled by the negative charge, and it gets into the cell finally. So it takes a while for water to tumble through the plasma membrane. We were doing a test experiment up in lab where we're blowing up red blood cells. A guy was doing that experiment in his lab, and he got to thinking, well, if water's moving so fast following these solutes that are lipophilic, if it's water that's lysing the red blood cell, how is water tumbling through the plasma membrane so fast that following the change in concentration due to propyl alcohol, you get lysis in 10 seconds or less? And so he thought there must be channels that water is able to go through. So he went out looking for water channels. He discovered aquaporins, and he got a Nobel Prize and $1 million. So simple experiments like you're doing in lab, if you think, what are the consequences of that? Why is the membrane behaving in this fashion? Maybe you're going to discover something and get a Nobel Prize and $1 million. You never know. Okay, so there are channels in the plasma membrane. They're set up by proteins. The protein structure is making an open area in this bilayer, and specific ones allow water to move, and others will allow ions to move. And then we're going to have other proteins that may be carrying things across the membrane. So we've got the things going across the plasma membrane. If you want to do large volumes, bringing in or taking out, you probably want to have what's called endocytosis, where the plasma membrane invaginates, and whatever gets into that area then is closed off in a vesicle, it comes into the cell. If you want to get rid, not rid, but if you want to release a lot of things, then package it in a phospholipid bilayer, confuse with the plasma membrane, and then open and release all those substances. So endocytosis, exocytosis of molecules, you can have these receptor-mediated endocytosis, and then you can have vesicles, exocytosis, and get these things across. Or you could have carriers. carriers are limited by the number of things they can bind and bring in. So if you want to bring a lot of things in, then endocytosis. If you want to move a lot of things out, exocytosis. If you want to put a lot of proteins in that will handle a few of these molecules in and out, you could do it that way as well. So receptors that will, in the membrane, that are triggering endocytosis means they're going to be bringing a specific thing in. Not everything is going to bind to that receptor, so not everything is going to be accessible to the inside with receptor-mediated endocytosis. Some cell adhesion proteins are also in the plasma membrane for the purpose of attaching cells to their neighbors or temporarily attaching cells for recognition purposes, immune recognition purposes. Enzymes are in the membrane. A lot of hormones that are out there that are hydrophilic can't get through a channel because they're too big. They can't get through the lipid part of the plasma membrane because they're hydrophilic. So they have to have a receptor to bind to. How does the message of that hormone get to the inside of the cell? You have the receptor linked to another protein on the inside that then stimulates an enzyme that will then activate this second messenger system inside the cell. It sounds like a whole complex scenario for very little effect, but it's actually exactly the opposite. One hormone outside can get several thousand times of the cell response that a lipophilic hormone going in would get. They get it faster, they get more, they get an amplified response inside the cell. The lipophilic hormone, you got to turn on the genome, you got to get things transcribed, then you have to translate, and then you got to release them. It takes time. So membrane-bound enzymes, some of them are, most of them are on the outside because they're enzymes that are involved in digestive process so that you get something small enough to get absorbed by the cell. So a lot of enzymes in the kidneys to break down things that are filtered so you can get them back and not have them go out in the urine. Digestive system, break them down so your food that you're eating can be absorbed for a nutrient. So glycoproteins, glycolipids. You stick a protein in the plasma membrane and then attach a carbohydrate, which is a string of glucose molecules on the surface that's branching probably. And this is a protein, this is carbohydrate, now it's glycogen because it's a string of glucose molecules. You have a glycoprotein. If this glycoprotein is attached to the phosphate head, this is branching out here, then you have a glycolipid. So these can be used as receptors. They can bind things on the surface as a receptor. They can be used as self-markers so that the immune system knows this cell belongs to you. If you transplant my cells into your body, your immune system will see my markers as foreign and attack it. That's what happens when you inject an organ in transplant. Also, contact inhibition. When these surface molecules come in contact with each other, it should signal the cell to stop dividing. You divided to fill in the gap, and now when you fill in the gap, cell division should stop. Some cells lose that contact inhibition, and then they continue to divide, and you get a tumor. So a benign tumor or a metastasizing tumor, those cells have lost their contact inhibition, and they filled in the space but continue to divide, and then they create problems. So the term fluid mosaic model goes back to, can things move in the plasma membrane? Things need to move in the plasma membrane. If we have hormone out there that's in high concentration in a certain area of the surface of a cell, you don't want to wait for the diffusion of the hormone to the receptor. You can have that occurring, but move the receptor to where the hormone is in high concentration. So get a motor protein to drag that receptor to the area where the hormone is in high concentration. So you want things to move so you can concentrate them in the plasma membrane. So the presence of the kinks in your lipid tails, that creates fluidity. Presence of cholesterol. Too much cholesterol, the membrane is rigid. Too little cholesterol, the membrane also is not as fluid. So you need just the right amount of cholesterol in the membrane for the ability for things to move. The mosaic pattern is the proteins that are moving. The movement of the protein is the fluidity that they're talking about. So here are examples of proteins. Anything that goes across the entire membrane is either referred to as a transmembrane protein, or it's in the membrane, so an integral protein. So integral protein here, we've got one that has glycogen on the surface, so glycoprotein. So it could be acting as a cell marker. We've got one here that is also a glycoprotein, and down here it says it's acting as a receptor. So we've got two types of glycoproteins. One is saying it's a receptor. It's linked to a protein that's on the inside of the cell. It doesn't go through the membrane, so it's referred to as a peripheral protein. It's on the periphery of the plasma membrane. These are linked usually to enzymes on the inside that then will trigger second messenger systems. So out here, the receptor binds a hydrophilic hormone, and then the second message is going to be generated by enzyme cascade inside the cell. This leak channel, it's a protein that is designed so that when the amino acid sequence then folds and forms the globular portions, you have hydrophilic amino acids here, hydrophilic up here, hydrophilic here, and then the core is all hydrophilic, and then hydrophobic amino acids in these outer regions. They're always open. They're always allowing whatever can fit through to go through. Goes through either by size or charge, selectivity. If an ion goes through a leak channel, water also is going through those leak channels. So we've got leak channels for potassium. Potassium goes down its concentration gradient out. Sodium goes down its concentration gradient into the cell. But you always will have high potassium in and high sodium out. So these leak channels are always allowing the ions to move. There must be something that isn't correcting for that leakage. So your cells will have proteins in the plasma membrane that will move sodium out and bring potassium back in. You have the leak passively happening, but this movement is up the concentration gradient. It's going from low sodium to high sodium. So sodium is going out, potassium's coming in, and it's up the concentration gradient. So this protein has to be using ATP and then phosphorylating the protein that's allowing for this transport. So ATP is becoming ADP. You're losing one of the phosphates in this process. So it requires energy for that to happen. Here we've got another protein. It's called a carrier protein. So cells may want to selectively allow something in or selectively put something out, but not have it happening all the time through a leak channel, not moving a whole bunch, so no endocytosis or exocytosis involved. You have other proteins that are, let's give an example of a molecule that is using the tendency for sodium to be in high concentration outside, low concentration inside. If sodium binds to this protein on the extracellular side, suddenly there's a binding site that appears for another molecule from the outside. So sodium's always there, so probably this binding site is always there. So the molecule that is co-transported with sodium is glucose. If you don't have high blood glucose concentrations after a meal, it's highly unlikely that glucose is going to find this transporter. But if you have high concentrations, then glucose will bind. And then the gradient for sodium to enter is there. So the protein, once you have both molecules bound, it changes its shape, brings both molecules in. Sodium diffuses off because it's in low concentration inside. The binding site for glucose disappears, glucose is released inside, and then the protein goes back to its original shape. It flops back out. So sodium and glucose get co-transported in by these types of proteins that have binding sites. So that one shows one binding site, but some have two binding sites. The microfilaments in here bound to proteins. The microfilaments interact with other filaments, and you have a little motor track on a little protein moving on a contractile filament from one area of the plasma membrane to another. So these are acting as movement molecules. Microtubules also can act for movement. Sometimes it's intracellular movement. Microtubules can also, if you have cells that want to move from one place to another, like your immune cells, they assemble in the direction of the chemicals that are attracting the cell, disassemble behind, and they're like laying railroad tracks. You take a track from here, you put it in front, move the train, then take that one, put it in front, move the train, take that one, put it in front. So you're constantly assembling and disassembling microtubules, and the cell will move wherever the chemicals are attracting it. Gated proteins, sometimes they're ligand gated, so something has to bind to the outside to open the channel. Sometimes depolarization of the membrane, so electrical charge, will open gated channels. But gated channels will not be open all the time. Something has to, either a hormone, neurotransmitter, or electrical charge change will open the channel, and then an ion will flow. Yeah. Sometimes it loads, but it doesn't work. So it's not that important to see the cell membrane picture. Since we're running behind and I want to sort of catch you up to the other class, you can look, if you have online access to the animations, look at it. If you don't and you want to see them, you can come to my office and I'll show them to you. Okay, cell adhesions, they're going to be linking cells to each other, maybe temporarily, maybe on a more or less permanent basis until a cell dies. But you're going to have collagen, elastin, and fibronectin to help to keep cells in place for their proper functioning. You can't form a tissue if the cells come apart. You can't have an epithelial tissue if it comes away from its basal lamina. So fibronectin will be in the basal lamina to help keep the cells put. Stay put, that's what fibronectin tells the cell. Stay where you're supposed to be, don't be moving elsewhere. Elastin allows for stretchability. Collagen, flexible but not stretchable. So you're going to have these various types of junctions between cells. They'll have various components of proteins. The desmosomes, we'll see, are the proteins. I don't know if somebody knew about desmosomal attachments and invented Velcro. Certainly your cells didn't see Velcro and invent desmosomes. So maybe they saw the interaction of the proteins in the desmosome. But then I think in my memory way back, somebody saw thistles and saw things that attached in nature and invented Velcro. So I don't remember. But if you look at desmosomal attachment, the interaction is like Velcro. You have fuzzy proteins in Velcro and or fuzzy plastic, and then a hook that you press in there and it attaches to the other plastic, and you've got the thing stuck together. The desmosomes, you have these cadherin molecules that are secreted by adjacent cells, and they hook up in the center and don't allow the cells to come apart. On the inside of the cell, the cadherins are attached to intermediate filaments, usually keratin, maybe some other other filaments as well. They're the most abundant intracellular attachment in the body. So if in doubt, you're asked about what cell adhesion molecule it is, I guess in lab, say desmosome, because it probably is what they're talking about. The others, you have tight junctions. Tight junctions are formed by other proteins. They're, as their name implies, they're supposed to prevent things going between cells. So you have these occludin proteins that come from each adjacent cell, and they attach to each other. It's referred to as the kiss site, which is sort of romantic. The location of the tight junction is towards the apical surface of epithelial cells. The transcellular pathway should be eliminated from the chance of particles or pathogens, other things that may be out here in the lumen, from getting between cells and then into your body. So tight junctions are designed to prevent this leakage. We're going to find out next semester. They get leaky, especially when a lot of absorption is occurring in your digestive tract. They become leaky. So down below, you're going to have a lot of immune cells hanging out to make sure that the bacteria that's out here isn't getting into the rest of your body. So tight junctions, apically located, they go all the way around the cell. What they're trying to do is force things to be selectively absorbed based on, do they have a channel, do they have a transport protein, is there a mechanism to get them across? Then if there is, the body must want them. Okay, gap junctions, as the name implies, there's a hole. You have the connexon subunit forming a channel between adjacent cells, so either electrical charge can go between cells, or nutrients or hormones can go between cells. So in the seminiferous tubules, when spermatozoa are developing, you want them all to develop at the same rate. So from one stem cell you get 4 spermatozoa. And you do that by sharing nutrients between adjacent spermatozoa and also hormones that drive the maturation. In hard and smooth muscle, the gap junctions are there for electrical stimulation of adjacent cells. So you have different purposes for those gap junctions. Okay, permeability is something permeable? Is it not permeable? I'm out of time, so gotta wait for next week, Monday. Probably still have time to understand your lab homework before you